Hello, Rasmus. Hi, everyone.
Hello.
I suggest to wait like maybe another minute. People are joining now. All right, so we have the second lecture by Professor Rasmus Nielsen from University of California in Berkeley today. So Rasmus, thank you very much for coming.
Thank you for having me. And let me start by sharing my screen. You need to enable it for me or make me co-host.
Yes, now you can share. Sorry.
All right, there we go. All right. So what I thought I would do today is to talk more about coalescence trees and coalescence processes and how to make inferences, mostly of demography, but we'll also be talking a little bit about natural selection at the end of the lecture. And I gave a brief introduction to coalescence theory last year. I won't spend a lot of time on that, but I will review some of the basic ideas about coalescence trees, just in case that people have forgotten, and to make sure that I have all the appropriate context for everything. We're going to start by considering the case of when they are just a sample of two individuals. So this figure, what that is supposed to depict is a population where you have the past of the population here and the present here, which then take a sample of two individuals. Each node is an individual in the population, and each edge here, that's a parent-offspring relationship. So this individual is a parent of that individual, for example. And then we can trace the population back in time, all the family relationships, who is the parents of who, back in time. And you can see this is simplified. It's essentially a haploid model where each individual only has one parent. In the past, this corresponds to, for example, a Wright-Fisher model that we use in population genetics. And each row here is then a generation. So you can think of this as a discrete generation model, where this is the present generation, that's one generation ago, 2 generations ago, and so on. So we take the two individuals that we have in our sample. All these blue individuals are individuals that are in the populations, but we haven't sampled them. And then we trace the ancestry back in time of these two individuals until the two of them have a most recent common ancestor. So we say then that these two lineages here have coalesced. And it induces a coalescence tree that we can write like this. A certain time in the past, these two lineages here, they coalesce, meaning that these two individuals have their most recent common ancestor down here. So this coalescence time, in this case, is the same as the time to the most recent common ancestor. So this is for a sample of two. You can see we have a little tree with 2 leaf nodes and 1 internal node here, root of the tree. In this rooted tree, and that's the coalescence tree for a sample of 2. Of course, we can do this for larger samples for any sample size n. In this case, it's an example with 4 individuals. We do the same thing. Each node here is an individual, and edge is a parent-offspring relationship. And we can, for the 4 individuals in our sample, we can track their ancestry back in time until we have these coalescence events. The coalescence events are the times at which, the points in this graph, at which 2 individuals for the first time have this same ancestor. So there was an individual here that had 2 offspring that eventually are ancestors of these 2 individuals today. So this is their first common ancestor. And we can track down then for all individuals and figure out when they have a common ancestor. So these 2 individuals, as I said, they have an ancestor here. And then if we also add this individual to it, we can see there's an individual here that's an ancestor for all of these 3 individuals. And then if we go all the way back to this time, we have an individual here that's the ancestor for all 4 individuals. So the coalescence tree we get is like this. All right, this node here in this tree corresponds to that individual here. The node here in the coalescence tree corresponds to this individual here. And the node down here corresponds to that individual there. So down here, we have an ancestor for all individuals in the sample, the most recent common ancestor for all of these 4 individuals. Okay, so what we talked about last year was that you can, under suitable assumption, you can generate probability distributions of these coalescence trees. And the logic for doing that is based on taking pairs of individuals, and then I don't know why I still have this bar. And can you see this bar moving around on my screen, or do you not see it?
Yes, we do.
And I don't know how to get rid of this bar. If anybody knows that, let me know. Usually, I think if I am, it's not there. It's there last time too, though. Anyway, I'll just move it around. So we can with a generative process forward in time. For example, a Wright-Fisher model, we can calculate what's the probabilities of having these most recent common ancestors, and we can come up with probability distributions for that. And I went through this slide last year, how to do that. So the major conclusion is that in a sample, when there are n lineages in the sample, if we look backwards in time, the waiting time to the next coalescence event is exponentially distributed, will mean equal to 1 divided by j choose 2 if there are j lineages. So if I go back to the previous slide, or maybe I can take this slide here, yeah. If we look at the previous coalescence tree here, the time to the first event, so from the present and looking backwards in time, the time until there are only 3 lineages active, the time until the first coalescence event, is exponentially distributed will mean 1/6. The time from their 3 lineages until there's the next coalescence event, so now there are 2 lineages, is exponentially distributed will mean 1/3. And the time from 2 lineages to 1 lineage is exponentially distributed will mean 1. So what are the units here of these? Well, these are in units of 2n generations. So when I say that the expected waiting time here is 1, that is really 2n generations. I mean, 1/3 here, that's 1/3 of 2n generations. So 2/3 n generations, where n is the population size, not the sample size, but the population size. So for example, if you take 2 individuals and look at a particular position in their genomes and ask what's the time until they have a most recent common ancestor in that part of the genome, if the population size is 10,000, you would expect to wait in average 20,000 generations. And the waiting time, that's a mean, and the whole distribution is an exponential. Okay, so that's under our very most simple process, coalescence process, the Kingman's coalescence process, that assumes that there's no natural selection, that there's random mating, and population sizes are constant. So just as illustrated here, the population size is the same in each generation. That's also an assumption of this model. So we get a sort of relative simple probability distribution of trees based on this, knowing that the waiting times are exponentially distributed with these means here. We can then write down probability distributions of different coalescence trees. And that we can then use for inferences. And that's really the topic of today's lecture, how to use that, how to use probability distributions on trees for inferences about the processes that generated the trees. One thing you, for example, here should already realize is that since they expect the waiting times here are exponentially distributed with some constant times the population size, you have faster coalescences. So that would be shorter branch lengths in the tree if the population size is small than if it's large. So you should already now realize, we can look at the waiting times here between these coalescence events, or equivalent the length of these branches, to say something about the effective population size of the population. Okay. And maybe I should qualify now by one more thing, that I use the word effective population size. That's because in real populations, and of course don't behave like idealized populations like this one. But an effective population size is then a concept population geneticists have invented to indicate the size of a population that would generate the amount of genetic variability that you see, or maybe the rate of coalescence that you actually observe in the real population. So if we look at a population of humans, we say the effective population size is 20,000. That means that there's an amount of genetic variability in that population that would be generated in an idealized population of 20,000 individuals. But the actual number of individuals in this population may be much larger. Typically, it's much larger than the effective population size. But the word effective here is sort of used to indicate that we understand that there's likely some discrepancies between the true model under which these populations evolve and then the idealized models that we use. So we often use that term, the effective population size. All right. Just a little bit about the words I've been using here to remind you about all of these words. I should say I write my trees a lot of the time like this, with a root down here and the leaf nodes up here. That's something that if you're a computer scientist, for example, you would never do, or mathematician. Usually they go either with a root at the top if you're a computer scientist, or maybe the root out to the left, but really like this. But since I'm a biologist, I like to draw my trees like this, with the root actually at the bottom and the leaves are up in the top. As a good biologist, I know that's how trees actually grow out in nature. All right, some of the words that we're using for about the trees. The leaf nodes here, we have nodes in the tree where the edges join. We have nodes. We talk about internal nodes and internal lineages. Those are ones not leading to leaf nodes. Leaf nodes are the ones that actually represent the observed data that we have. Those are the leaf nodes in the tree. We can talk about external lineages leading to a leaf node and internal lineages that don't lead to a leaf node. And down here, we have the most recent common ancestor. As I mentioned before, that's the ancestor of all the individuals in our sample, not necessarily in the population, but for the sample. All right, so I'm going to give you some questions today. And the first question I'm going to ask you is, how many rooted trees are there in a sample of size n? So for an arbitrary number of leaf nodes. And what do I mean by number of rooted trees? Perhaps best explained, you would need to know what I understand by isomorphic trees then to be able to answer that question. But it's probably best explained by an example here. Here's a case with 3 leaf nodes. So n is equal to 3 here. And for this particular case, there are 3 different rooted trees. A first join with B and then joins with C, or A first join with C and then they join with B, or I may actually made an error. This should be B joins with C here, and then A should be out there. I switched the order here of these ones. So this should be A joining with B, and then the C as the outgroup. The one that is the most distant one is called the outgroup. So that should have been A here. So this branch, imagine this branch is going over here. All right, but so for n equal to 3, there are 3 unrooted topologies. And the question to you is, how many rooted topologies are there? How many different rooted trees, if you don't consider the branch lengths, are there when n is equal to 3? And I'm going to, I like to maybe, it's not a big class, there are only 14 participants, but let me send you out in some breakout rooms. Am I host here, Vladimir? I don't think I am.
Excuse me?
Am I host?
No, but I can do the host.
You can make me host. I can put people out in some breakout rooms. And so like, I think then you can discuss this problem in Russian. And like, I'm going to send you out in like 3 groups, and then each group should come up with a a solution to this problem. All right, so let's see. Why don't I see breakout rooms here? Oh, all right. I'm destroying everything here by already giving you the solution. Breakout rooms, there we go. I'm gonna send you out in 3 breakout rooms, and then feel free to discuss in Russian the solution to this problem, and then I'll pull you back in 3-4 minutes from now. So please, if you haven't joined the breakout rooms.
Yes, sir, I didn't answer.
Yeah, you didn't. That's good.
I just
okay.
Got like reviews for our
No,
paper after 4
yeah.
months, and I was reading it. It's about our simulator, so
Okay.
It's plus computational biology, and they suggest like to resubmit it as a software paper.
Mm-hmm. And how are the reviews?
Well, it's like I think they like it. They just say that it should be like presented as a software paper, not like as a method paper, because we do not make any data analysis. And they also want some like comparisons against some particular simulators in R.
Other simulators and so on, yeah.
Yeah, which we ignored. But do you think is it better to proceed with PlusCom Bio or just make a short application note for bioinformatics?
I mean, if you get in plus computational biology, that would be good, I think, right? I mean, if they open up for submitting a revision, I would always do the work and try to submit the revision.
Okay.
So I'm gonna go to the bathroom for a second, and then I will bring people back when I'm back. We'll start again in about half a minute when everybody has been brought back. All right, welcome back. So let me hear what you find out. So we have 3 different breakout room. We could, for example, take room 3 first. That's a room with Anastasia Mikhailova and Kirill Alexeyev. Sorry about my wrong pronunciation of your names, by the way. But maybe that room, did you come up with any solution?
Well, we were thinking about solution and about how we can combine the leaves and how we can write it mathematically, but we haven't come to a conclusion exactly. We have some thoughts about it, like let me think how to explain it. To take into account the possible positions of those leaves and the possible ways to construct a tree, like to make where would we place a node.
Yeah, I mean, I think that is sort of the logic that you need, and but it takes a little time to develop. Let's hear for some of the other group. For example, the group 2 with Dmitry Sirotkin in it, and Victoria Vasileva. Did you come up with any solutions?
Well, I've come up with an intuition, not the solution. Wasn't able to take an exact formula. I'm pretty sure the number of set trees is well exponential, yeah. And the naive approach is like, let's take first 2 leaves and then next 2 leaves. They were unsuccessful for obvious reasons, because there are symmetrical trees where the order doesn't matter. I came up with an idea that basically each rooted tree is a number of, well, let's say, separation of subsets of the vertices by 2. Yeah, when you take the very root and you take 2 main branches, that basically means that you separate the set of our vertices.
You can derive it like that, but it's a very hard way of doing it.
Yeah,
but it can be done like that.
Yeah,
there's a trick for this that maybe, I don't know, maybe I'll hear from room 1 if they came up with anything.
Yeah, wasn't able to get the formula, but I put in that direction, try to do something. Yeah,
absolutely. Room 1 with Dmitry Lisin and Galina Vlasenko and Leo Planchet. Did you come up with anything?
I don't think we did. I don't know. I don't speak Russian, so I'm not sure, but I didn't come up with anything. I think that I misunderstood the problem. Because you have to remove isomer. So in this example, there's only 1 tree, right? You need to remove isomorphism.
They're labeled, the leaf nodes are labeled. So that's the thing. So you have a labeling of the leaf nodes. So these 3 trees are all different from each other, except for the error I made for the 3rd one. Well, sorry about saying that you should do it in Russian when there was a non-Russian speaker. I forgot, sorry about that. I just thought people were more comfortable if it was in Russian. So the easiest trick for doing this is to think about how many different topologies is there for 2. Then what if you add 1 more species, 1 more leaf node, how many are there for 3? And what if you add 1 more and so on, and doing that recursively. So for 2 leaf nodes, you just have 1 tree. There's just that 1 tree. So whether I write it BA or AB, they are isomorphic to each other, the way I define it. That's just 1 tree. For 3 leaf nodes, there are 3 different trees. And one way to think about it is that when we add species C, we can add it either on lineage A, on lineage B, or on the root lineage that's not drawn, but would be here. So there's 3 different ways we can add C to generate 3 different topologies. Okay, so now we do the next trick. The next time when we go to 4 leaf nodes, we now add D, and D can be added in 5 different places. 1 here, 2, 3, 4, and then on this as a root here. That's the 5th one way to do it. And you realize that every time that you have that there's a general rule for that when you have free leaf nodes, there's you can add in 5 different ways. When there's the 4 leaf nodes, you would have to be able to add in 7 different ways, and so on. So there's a simple rule here that every time you add another tree, you can go in 2i minus 3 more ways that you can add a root, add a node, if there currently are I lineages in the tree, or I leaf node. So you come up with this simple expression here that it's the product from I equal 3 to n of 2i minus 3, for which there's this identity here. It turns out that this product here can be written like this. But the way to think about it, the easiest way to think about it is just setting up a recursion of how many, every time you add another leaf node, how many new different topologies how many different ways can you do it that leads to different topologies. Okay, so that's the trick for that, and the general formula for the number of topologies down there. So it's a large number. You can see it has that factorial there. It's a very large number. Okay. All right. So that's the number of trees, and I think that's a good exercise just to start thinking a little bit about structures of trees. So I like to give those kind of exercises because it gives you a sort of a time to think a little bit about what a tree is. I should also, there's certain things that didn't quite define what kind of trees. We're only looking at binary trees, for example. I didn't tell you that. I did that last year when I talked about coalescence trees, but these trees are always binary trees. That means that each node has degree 1 or 3. That is that it connects either just to one edge if it's a leaf node, or to 3 edges if it's an internal node. It's a binary tree. All right. So this is the genealogical history of a sample, the history of how individuals are related to each other. But how is that then related to the data we actually see, which is DNA sequencing data? Well, the simplest assumption that you can do to relate it to the DNA sequencing data is to assume that mutations occur at a constant rate according to a Poisson process along each lineage. And then to make it even simpler, we can assume that each new mutation makes a new variable site. That's what's called the infinite sites model. So every time you have a new mutation, you get a new variable site. So you can, if there are 10 mutations in the history of your sample on the tree, then there will be 10 variable sites. So I put some mutations on this tree, these little red explosions here. They are places in the tree where there was a mutation. And you can think of it as times in the tree where there was a parent. When the DNA was copied from the parent to the offspring, an error happened, so the offspring is different from the parent. And then if we have that will induce some sequences like this. And these are binary sequences, so you can just replace it with nucleotides if you want. Say 0 here in this site is A, and 1 is T for the next site. 0 could be G, and 1 is C, and so on. But because of this infinite sites assumption, there's only 2 nucleotides at most in each site, because you can never have a mutation that hits this, there can never be 2 or more mutations hitting the same site. Because this infinite sites assumption, remember, says that each new mutation induces a new site. So it's always biallelic. There are only 2 alleles, and therefore we can just write it as a binary sequence. It doesn't matter if it's AC or TG or whatever it is. And the way this relates to the tree is, if you look at the tree here, there's a mutation that happens on this lineage here, leading to the 3rd sequence. That's the first variable site. Here, everybody else, 1, 2, 4, 5, and 6, they have the ancestral states. Only 3 carries a mutation. So 3 get a 1, and the other sequences are 0. And we look at this mutation here that happened at the ancestral lineage, leading to both 1, 2, and 3. So that means that all of these 3 lineages, all of these 3 leaf nodes, these 3 sequences will carry that mutation. But none of these ones will carry that mutation. So that's the second side. Here you can see 1, 2, and 3. Those 1, 2, and 3 here, they carry the mutation. They're 1s. They have this mutation. 4, 5, and 6, they do not have that mutation, and so forth. For example, here there's a mutation that happened on the ancestor lineage to 4 and 5. So 4 and 5 have the mutation, and none of the other ones have it. Here there's mutation that happened on the ancestor lineage to 4, 5, and 6. So that's this side, 4, 5, and 6, they have the mutation. 1, 2, and 3 do not. And then finally, there's a mutation here that happened on the lineage, on the edge in the tree, leading to sequence 6. And so only sequence 6 has that mutation. So in this way, we can connect the genealogical process, combine that with a mutational process to make a process that generates data. And so, in fact, if you were to simulate DNA sequencing data under a standard neutral coalescence model, the sort of standard model of generating genetic variation, the way you would do it is you first simulate a tree, for example, like this tree, and after that you simulate mutations that you lay down along the lineages according to a Poisson process with constant rate. And that rate, we call that the mutation rate. And that generates sequences. That's how we approximate the infinitely complex pattern at which individuals have offspring and mutate and so on. We approximate that with this relatively simple process. An important thing here is that we can decouple the genealogical process from the mutational process. And that is true. That's always true. You can always do that, except if there's natural selection. If there's no natural selection, we can decouple these two processes. But if there is natural selection, that essentially means natural selection that whether you have a mutation or not affects how many offspring you would get. And in that case, you cannot decouple the mutation process from the genealogical process, because the genealogical process depends on which mutations you have. But that's only if there's natural selection. And you can think of what is natural selection. You can think of it as when you can't decouple the genealogical and mutation process, then there's natural selection. If those two can be decoupled, then there's not natural selection. So it's almost a definition of what natural selection is. All right. So now we're going to get into inference. And briefly, I derived something, some estimators last time of effective population size we talked about last year. And you're all forgiven if you can't remember that or if you weren't there, but ways we can just with simple summary statistic estimate this population genetic parameter, which is called theta, which is 4n mu, the product of population size and mutation rate. Okay, so in almost always, or for these models, always when there's no selection, these two parameters, the mutation rate and the population size, will only appear in any likelihood function as a product of each other. So we can't independently estimate these two. So instead, we estimate their product. And for historical reasons, we use this 4n mu, which is theta, as what's called the fundamental population genetic parameter. It's equal to the expected number of pairwise differences. So you can use the expected number of pairwise differences as an estimator of theta. That's one thing we talked about. You can also calculate how many mutations you have, and then relatively simply calculate for this Kingman's coalescence process, the standard coalescence process I talked about, how many expected segregating sites, that is variable sites you have, how many you would expect to see, and relate that to the observed number, and from that generate an estimator of theta, again based on S, the number of segregating sites. So these are two different ways that you can use summary statistics to estimate theta. Of course, that's fine and good, and people do this all the time, but of course, usually in population genetics, we're interested in much more than just this parameter. And we also realize that there's a loss of information by just using the number of segregating sites or the average number of pairwise differences, that we throw away some of the information and data by doing that. And most importantly, it's a little bit boring because we're just estimating that one parameter. What we're really interested in is something much more complicated. We're interested in how populations have evolved from each other, what their population sizes are through time, how much migration there is between populations. And you can then, to get at those questions, extend the coalescence models to many other scenarios. And so here's one example. I talked about this last year, as an example of a more complicated model. Here we assume in this model that back in time, there was an ancestral population, but then this population split up to two different populations. So you can think of this as maybe different species, there's speciation, or just two different populations that sort of separated from each other why do populations separate from each other? Hosts are different reasons, but some individuals swim out to an island and then they evolve independent out of that island. You can think of it like that, or suddenly a volcano erupts and separate the territory into two. Instead of having one population, you now have two populations. Those are some extreme cases. In real life, there's an infinite of different ways and processes that can cause populations to split up. But in the simplest case, we can model it like this. We have an ancestral population split up to two populations. We can then imagine taking samples from these populations. So for the second population, we take two individuals here. From the first individual, we take two individuals here. And then we again trace their lineages back in time and find the most recent common ancestor of these two individuals here. These two individuals that don't have a most recent common ancestor while still in this population, their lineages trace their ancestry trace all the way back to this population. So back here, we have a coalescence event that happened between individuals, an individual from population 1 and the ancestor of these two individuals from population 2 that happened here, that coalescence event. And then all lineages finally coalesce down here. And that then gives us a coalescence tree like this, with the two individuals from population 2 here and the two individuals from population 1 here. Now, the major point is that we can define, again, these, write down mathematical models for this. And they're not that complicated usually. It's relatively easy to write down these models. So that means we can write down, then also start writing down probability densities, probability distributions of trees. Okay, and now these trees will depend on some parameters we're interested in. For example, in this model, we might be interested in what are the population sizes here of population 1, population 2, and the ancestor population. They could reasonably be assumed not necessarily to have the same population sizes, and might be interested in what those are. And maybe more importantly, what's the divergence time between the populations? That's a very typical question we're interested in. How long time since they split off from each other? So now we have 4 parameters suddenly. And we can write down a distribution of trees, of coalescence trees, as a function of these parameters. So as we change these parameters, that changes the distribution of trees, gives a different probability distribution of trees. So if we sample trees, if we see a tree, that will tell us about what parameters are most likely, because it tells us something about which distribution we have sampled that tree from. Okay, so that's sort of at the core of thinking in much of population genetics on how to estimate parameters, that the information in the data really comes from the tree. And you can show that in the absence of natural selection, all of the information in the data goes through the tree. So in other words, if we knew the tree, what the coalescence tree was, there would be no information in the data at all. That the tree would be a sufficient statistic for any of the parameters we're interested in. And it contains all of the information that would be in the data about the history of the populations. So even if we had, if we know that this is a true tree, even if we had 100 million nucleotide sites to look at from this individual, this individual, this individual, and that individual, all those sites wouldn't provide us any information. All the information is in the tree. So one way to think about this is that we're really only using the data to learning something about the tree. And the information about the history of the population is in that tree. All right, so that way of thinking leads to what some people call Felsensteins Equation, which is a way to write down or think about the likelihood function for demographic parameters. So here, we think of X as being DNA sequencing data, very generally. But let's first think about, for example, some mitochondrial DNA sequences from humans. And theta here is a vector of parameters we're interested in. For example, migration rates, divergent times, effective population sizes, and so on. All of these parameters of the generative model, the parameters we're really interested in. And we can write that likelihood in the following way: as the density, the probability of the data given the genealogy. That's the coalescence tree. So G here is the coalescence tree, the genealogy, times the density of genealogies given theta. We can decouple these two. And notice here, this does not, that was what I was arguing before. You can show that rigorously mathematically. I haven't done that, but you can, that this here does not depend on theta. Okay, and that's because of this decoupling between the mutational process and the genealogical process. Because you can decouple those two, we can write the likelihood like this. So it's as an integral or all genealogies, so genealogies that live in a space of all possible topologies and all possible coalescence times for all of these topologies. Okay, so you can think of this as a sum of all these discrete topologies that might exist, the number of which we wrote up before, and then a multiple integral of all the different coalescence times. Okay, so obviously this is hard to evaluate. We can do it for a sample size of 2, for example, and we can enumerate and do it for a sample size of 3, and we can also do it for 4 in many cases, but it doesn't really generalize. It's not something you can do for 20 sequences because there's just too many sequences, and we'll have, solving these multiple integrals for all of these different possible topologies just can't be done. Okay, so that was sort of the major question in population genetics, and to some degree still is, but it was a question that was really posed in the 90s by Felsenstein and by other people. How do we solve this computational problem? How do we approximate this integral? And the idea is that to use various approaches that sample genealogies. So you can use sort of stochastic integration techniques here, where stochastically we sample some. We don't have to actually look at all possible genealogies. We just have to look at some of the best genealogies, and then we can approximate this in various ways if we sample genealogies. Okay, so one thing you could think of doing is, for example, let's sample genealogies from this distribution here. You could think of that of the prior distribution of genealogies, and then for all the genealogies we sample, we then evaluate this function here, and we say, and then take the average of that. That should, as the number of genealogies we sample goes to infinity, converge to this integral. That is one way to do it. It would just be incredibly slow to do, because the chance of hitting in randomly sampled genealogies is something where this is actually large. It's very small. So we have to have better ways of sampling genealogies that's more informed, that's more proportional to this product. We want to be able somehow to sample genealogies that are proportional to the product of these two functions. And if we could do that, then we efficiently could evaluate this integral. Okay, so the first ways that was done, and it's still being done in many cases, is to use Markov chain Monte Carlo. Okay, so in Markov chain Monte Carlo is a way to sample from a distribution in cases where that distribution is only known up to a scaling factor. So in this particular case, what we want to do is we want to set up a Markov chain which has state space on all the possible values of theta and g. So genealogies, coalescence trees, and values of the parameters. We're now going to assign prior distribution to our Bayesians and assign a prior distribution to these parameters. But the same thing can also be done in a non-Bayesian framework. But this is the easiest way to think about it. So we make a Markov chain on this state space of possible genealogies and parameter values. And we wanted to have this stationary distribution, the posterior distribution of parameters of genealogies, which is proportional to the probability of the data given the genealogy times the density of genealogies given the parameters times the prior distribution of the parameters. It's proportional to that. So we can use Metropolis-Hastings sampling, for example, to set up a Markov chain that samples from that by proposing changes to the distribution that to the current state. You're currently in a state, where of theta and g, and you want to go to then theta prime and g prime. You propose a change to the current state, and you accept it with this function here, which you can see what you have is, well, I'm assuming here that P of theta is uniform, so that cancels out. So you have a term here, this function here times that function, and then you correct with this ratio of proposal probabilities of going from the new state to the old state divided by the probability of proposing a change from the old state to the new state. So that is standard Metropolis-Hastings sampling that you may or may not be familiar with. And I think if you haven't seen something like this before, this is probably not enough for you to really understand what's going on, but the ones that have seen MCMC before should be able to realize that you can sample from a distribution like this. And when first you do that, then you we can approximate that integral, in particular the posterior distribution. Then you get a theta. If you put in uniform values for theta, is exactly the likelihood function here. Will be proportional to that. So now you can evaluate the joint likelihood function for multiple parameters and get the likelihood function like this by simply sampling values of theta from this distribution. All right. So if this doesn't really make sense to you because you haven't seen this before, I would encourage to read up on literature on Markov chain Monte Carlo and that. But there's a standard trick that we're using here from simulating from a Markov chain. So we can get a stationary distribution that in this case is proportional to this function here. All right. Now consider it as a function of theta. We consider this a likelihood function that we want to use to estimate theta for. So, okay, yeah, maybe I should say that also. Why in the first place are we interested in this function? This is, of course, the likelihood function for theta. We maximize this function for theta, and then we that gives the maximum likelihood estimates of theta. So that's why we're evaluating this function in the first place. So the take-home message is that this very complicated integral of sampling that you have to evaluate, sample all these genealogies and solving this multiple integral over coalescence times can be done numerically by making a simulation approach that essentially simulates from this distribution. If you now consider this as distribution of theta, something proportional to that. Okay. And then if you take the maximum in that function for theta, then that will be the maximum likelihood estimate of your parameters. All right, so that's the take-home message. So how do you actually do that? Now you have to sample genealogies. Well, what you do is that you represent the coalescence tree in the computer memory, and then you do various perturbations of that. You might, for example, move an edge around and reconnect it another place in the coalescence tree. And you keep proposing changes like that, updating them, accepting them with this probability, and that will then, in the end, you will have be able to sample correctly from the posterior distribution. If you do that, of course, this can also be computationally intensive, but it's been the background for a lot of different work on how to estimate parameters in population genetics. So here's an example. I think the first time this was done, some of our work from many years ago, we looked at some sticklebacks, so some fish that lives in the Pacific Ocean here, and you find them both in the Eastern Pacific and the Western Pacific. It's a little odd because they only live in coastal waters. They don't go in the open ocean, but they seem to be related to each other. And so researchers were interested in, are they coming from east to west or west to east, and how much are they migrating back and forth? Okay. So we took a model here in which you assume that two populations, the eastern and the western ones, they diverged from each other T generations ago. There's migration in one direction, there's migration in the other direction. And then you have these three different population sizes you also have to estimate. So you have to estimate 6 parameters. And then you can do this MCMC approach to this problem. And from that, you will get likelihood functions for the different parameters. This is a profile likelihood. So maximized over the other parameters for the 2 migration parameters. All right, so what we see here is that there's one likelihood function like this and another likelihood function like this. So I believe, as far as I remember, this is from west to east, and it's maximized at m equal to 0. That means that stickleback, they don't swim from west to east. Our best estimate is that the migration rate is 0. But if we look from east to west, we see that there is a value of about 0.5 or maybe 0.45, where the likelihood function is maximized. And that shows you that the data tells us that the sticklebacks are in fact swimming from east to west because the likelihood function goes down here, and you have a much lower likelihood if m is equal to 0 than if m is equal to, for example, a half. All right, so we get a maximum likelihood estimates of these parameters, of one migration parameter being about half and the other one being approximately 0. Okay, so we can now in this way solve these problems, trying to infer which direction have the fish been swimming. All right, so that's an old example. Here's a more recent example from some other software. It's not software we made, but other software where they're looking at Y chromosomes in humans and empty DNA in humans. So mitochondrial DNA and Y chromosomes. And what's going on here is that they are estimating effective population sizes using an approach like this back in time. So basically what in this kind of approach, you assume that there is a piecewise linear or piecewise constant effective population size, but then you allow, you integrate over all the ways you could make those piecewise linear times. So you can sort of smoothly estimate effective population sizes through time. And if you look at the empty DNA first, we see here, this is about 50,000 years ago, 10,000 years ago. And you have had this really, really strong increase in effective population size all over the world. For some reason, it sort of seems to be going down a little bit. There might be some artifacts that makes it go down a little bit, the empty DNA, very recently. But otherwise, we have had very strong growth in human populations, you see from many different places in the world over the past 75,000 years. Now, if you look at Y chromosome, we kind of have the same pattern, except that this massive dip here. You can see about 5 to 8,000 years ago, you have this massive reduction in effective population size in many different populations around the world. So why do you see it here and not here? Well, this is something that must happen particularly to men, to males. And there are several different hypotheses about why it is. And one is that you change social structure. So suddenly many places in the world, this is, associated with the introduction of agriculture, for example. So you had a completely different social structure. And it's also associated with increased mobility. And the thought is, what happened is that men, you had a social structure now where one man could have many, many offspring, that could have many, many offspring, that could have many, many offspring. That increases the variance in offspring number among males, but not among females. And you can think about, for example, Genghis Khan, which is known to have had 500 sons or something like that. And each of those sons, they became, a local chieftain in a local area, and each of them had 100 wives and had hundreds of sons, each of them. So in a social structure like that, one person, because wealth is inherited, and wealth is associated with a number of offspring, you suddenly can have this massive variance in offspring in males and not females. So that might be what's going on here. We don't know, but that's a hypothesis. We think that that's what's going on. But the fact is that there's something really odd going on in the Y chromosome. And we don't think it's natural selection because it happened in all of these different lineages all over the world at the same point in time. There's no way you would have to have 10 different mutations that suddenly happened independently, 10 different places in the world to be able to get this. It's much more likely it's through the spread of agriculture, which happened, and the change in social structure that happened at about the same time, many places in the world. That's at least what we think. It's a bit of a conundrum still. All right, that was just an example of an application of these kind of MCMC approaches to this kind of for inferences. Let me see if I'm spending too much time. Yeah, maybe a little bit too much time. Here's another example. This is looking at a bunch of different species of animals, some that went extinct, like the woolly rhino and the woolly mammoth, and some that didn't, like the horse and the reindeer. All around the animals that lived around the Arctic Circle over the last 50,000 years. And then what we did here is take ancient DNA from mitochondria and then estimate through time effective population sizes of these different animals and see, to see what happens when they went extinct and when they change population sizes. Now, these type of approaches, there's some limitations here in these computational approaches. They're also not so flexible. It's very hard to, to implement new models here. But so there are many other approaches people have been using to sort of approximate this kind of thing. And one of them is what's called approximate Bayesian computation. It's one of the more popular methods. So you're trying to approximate what we're doing here, but you approximate it by taking some simulating data and calculating some summary statistics. So the basic idea in approximate Bayesian computation is that you draw values of your parameters from some generative distribution, okay, that you know. And you're then interested in finding out, in some sense, which distribution is best at generating my data. But you first draw some parameters from that prior distribution of your parameters. Then you simulate data from a distribution given by those parameters. And then you reject that value of the parameters if the distance between the observed data, which is y here, and the simulated data is larger than some value epsilon, if the distance between the two is larger than that. And S here, that it's some summary statistic of the data. You have to be able to define as a distance. And to do that, you have to reduce the dimensionality. And so people come up with various summary statistics. And ultimately, what you're after is calculating some kind of distance between the true data and the simulated data. And by doing that, it's easy to show that as epsilon, if you choose epsilon going to 0, in that limit, what you will get, this will become an exact Bayesian computation based on this summary of the data. All right, and the larger epsilon is, the fewer simulations you will have to do, but the more of a rough approximation it is. There are also various regression approaches that people are using these days, so they don't use that sort of fixed cutoff epsilon, but that they use some more sophisticated approaches. But the major take-home message is what you're doing here is that you're trying to basically simulate data. And then you ask, okay, does my simulated data, does that look like the real data? And if it does, then you think that the values of the parameters you simulated under your data, your data under, that they might be good values. Okay, but if you simulate some data that's very, very different from the real data, then you don't believe that the model you simulate your data under is a good model. So in approximate Bayesian computation is more of a formalized way of saying, okay, let's find models to simulate data under so that our simulated data look at like the real data. And it can lead to this, you could formalize it like this, but that's basically all it is. It's sort of blindly simulating data, hoping to find models that can generate data similar to the ones you observe. So for the same problem as we saw before, we did that for some of these ancient and modern data, for some of these species, to try to estimate some of these models, to look at population sizes through time with discrete changes in population size, to try to figure out when did the population size start to change of some of these species. And this is the result we happen to get in these particular cases of these effective population sizes. And this roughly resembles a lot of what we saw using the MCMC approaches. And one of the major take-home messages is there wasn't a slow decline of the species. We don't see any evidence of that in the genetic data. It was a really abrupt decline of the species that went extinct. And it's not really easy from the effective population size back in time and trajectory of effective population size to predict which one goes extinct and which one does not. That was part of the conclusions of this paper. All right, that was approximate Bayesian computation. Back to the MCMC approaches. Today, there are several different MCMC approaches. The most famous one, most used, is probably BEAST now, that can do some population genetic models, and people use that, for example, for many of the viral analysis to do, when you analyze the history of SARS-CoV-2, BEAST is the most commonly used program to do that with. Other program, the various program called the IM programs by Jody Hay and myself have been developed those, and Migrate program by Peter Bierley and Joe Felsenstein that are implementing these type of approaches using Markov chain Monte Carlo to try to estimate a likelihood function. All right, so that was MCMC. Now, what you're doing here with MCMC is that, these approaches, is that you integrate over a tree. And I've been treating this problem as if there's only one tree. And that's true for mitochondrial DNA because there's no recombination there. It's also true for the Y chromosome because there's also no recombination for the Y chromosome. But of course, in nuclear data, in the vast majority of data we have these days, there is recombination. And the consequence of recombination is that you will have different trees in different parts of the genome. So you will have millions of trees in the genome. Each position in the genome might have a unique tree. There are millions of trees. Sort of a fundamental computational problem. These kind of approaches that I talked about so far that are great for mitochondrial DNA or viral sequences or Y chromosome DNA doesn't really scale up to whole genome analysis. So when we started getting whole genomes, 10, 15 years ago, many of the methods we had really were not appropriate. They could not scale up. When I was in graduate school, we always thought, okay, computational power is increasing so fast that even though we will be able to have more data in the future, the methods we develop today will certainly also be applicable in the future when we have more data because the computational power will increase faster than the amount of data we have. But that turns out not to be true. With the sequencing revolution in genomics, you actually had a much stronger increase in the amount of data than you had in computational power. So we can't, for example, people are using these type of approaches by chopping up the genome to a number of chunks, maybe 100 chunks, and then assuming that there's in each chunk just one coalescence tree, and then applying these methods on these 100 different chunks and multiplying the likelihood functions together. It's possible to do, but it doesn't work very well. It's very slow. It's only 100 chunks of the genome. And in most species like humans, recombination rate and mutation rates are on the same order of magnitude. So that means every time you have a new mutation that provides some information, you also in average will have a new recombination event. And so there aren't really chunks of the nuclear genome where you have sort of the same tree in large chunks. There's not much of that in the genome. And if you just particularly select out regions of the genome that seems to have that feature, you're probably biasing yourself a lot. So I would say that these methods that many of us spent a lot of time to develop in the 90s and the beginning of the 2000s, really in the age of genomics, are less applicable. Although there are all these problems about mtDNA and Y chromosome DNA and viruses they can still be applied to, they can't really effectively be applied to the whole genome data. All right, so let's think more about then this process of recombination and how it creates different genealogies. And I'm going to use this figure that one of my students, Deborah Brandt, just made a couple of days ago for a paper she's writing on to try to explain how we think of this process. All right, so we start with figure A. What you have here in gray is a pedigree. If you ignore all the blue and reds and black first and just look at the gray, you have a male here and a female. They make two offspring here. This female then make an offspring with that one and so on. And then their chromosomes. You can think of it, you would take some chromosomes and put in them, and then we track in this pedigree how they segregate down in the pedigree. Okay. And is being passed on to the offspring. And what we can see, for example, if we take this female here, there's a chromosome here and a chromosome here. And this X indicates there's a recombination event that put this red segment together with this red segment here. So now there's this red chromosome here. Okay, recombination events happen every generation like this and will shuffle around the DNA. All right, so we can sort of in the pedigree forward in time track these segments of DNA, how they've been passed. Or we can take a coalescence perspective and track it back in time, how the DNA is being passed back in time. So for example, we can start here, like before, we take some DNA here and we track it back in time. There's some other DNA here, and a coalescence event happen here between these two copies of the DNA. They have an ancestor here. That's just 3 generations ago. So it can fit. It's a very odd little small population here with a lot of inbreeding, so it can fit in this figure. But this way we can track these coalescences. But the new thing now is that we also have recombination events, and we can now track it back in time. So what happens back in time here when there was a recombination event here is that the DNA that's on this chromosome gets split into 2 chromosomes here. This one and this one. And then they have different histories back in time. They will segregate differently back in time. This one here, this chromosome then coalesces over here with this one here. All right, so the first part of the chromosome has the most recent common ancestor with this chromosome over here. But the second part then go back in time, split up again. There's another recombination event. All right, so now there's two here, and then recombines or coalesces back with this, the blue one and the green and the yellow one at 2 different times here, those pieces of DNA. So we can represent that as what's called an ancestral recombination graph. Okay, we have our 4 leaf nodes here, 1, 2, 3, and 4, corresponding to yellow, green, blue, and red here. And then we track now, in addition to having coalescence events as before, we now also have recombination events that leads to a split. So here there's a split, that's that recombination event. This lineage gets split into 2. Here's another split, that's this recombination event. This gets split into 2. There's coalescence there, there's coalescence there, and there's coalescence there. And now we have to then track on this graph which part of the DNA is represented on each edge to get the full ancestral recombination graph. We can think of also as a recombination graph, an ancestral recombination graph, as a tree sequence. If we go along the length of the chromosome here, from the bottom to the top, we first have a tree, a coalescence tree that looks like this. Now we're following this piece of DNA that's here. It coalesces with the blue one here. So that's this coalescence event here. We have that coalescence event here. That's this one here. And then this coalescence event here. That's this one here. And that induces then this coalescence tree. But then as we go further up the chromosome, we then have a recombination event that happens. That's indicated here. And that takes us to this tree here. Now we are following this piece of DNA that goes here. And then it goes over here. And then it coalesces here. That's that coalescence event here. Okay. Now we can follow the last part of the chromosome. And that goes over here. And then it goes over here. And then it coalesces all the way back there. So now we get a tree like this. There's a recombination event here corresponding to this recombination event here in the pedigree. And that takes us from this tree to that tree. Now, what you might realize is that these 2 topologies are actually the same. Like these are 3 different coalescence trees because the coalescence times are different. The branch lengths or the length of the edges here and here are different from each other. But they're the same topologies. These are isomorphic topologies with these 2 here, with leaf nodes labeled. These 2 are different from each other when the leaf nodes are labeled. All right. So if sometimes when we talk about tree sequences, what you'll often see is that you just distinguish between the changes in topology. So that's not the entire ancestral recombination graph. It doesn't have the information about this recombination event here. It only has the information about this one here, but it represents these 2 trees. Okay, so if we previously, the problem that we had was to integrate over a single coalescence tree. Now the problem that we have, the inferential problem that we have, is if we want to get the likelihood function, if we want to get all of the information out of the data, it goes through all of these trees, the entire ancestral recombination graph. And in real, in humans, there might be, in a typical sample, some million different trees. It'll be a sequence, a tree sequence of several million different trees. Okay, that's how many recombination events there would have been in the history of humans in the sample. That would have been thousands and thousands, in fact, millions of them, generating millions of trees. Okay, so that's a hard problem. This is a very, structure. You can imagine now you have 10,000 leaf nodes and you have 2 million trees that all are correlated with each other in a complicated correlation structure. That's a very hard problem to do. We can't do it with these MCMC approaches as before. It's just not possible. It won't scale up. So what do people do? Well, they look at summaries of the data. We already talked about summaries before using approximate Bayesian computation. And that's one thing people are doing. So you can calculate various statistics of the data and apply approximate Bayesian computation. But you could also do things that are a little more sophisticated in trying to get a little bit more information out of the data and do it more computation efficiently than just by these brute force simulation methods. And one of them is to summarize in terms of what's called the site frequency spectrum. And we talked about that last year, and probably all of you forgot what the site frequency spectrum is. So I'll just remind you. So in a sample of individual, the site frequency spectrum can be summarized in a figure like this, that gives the number of SNPs where the derived allele has a certain frequency. So on the y-axis here, you have the number of SNPs. That's the number of positions in the genome that looks like this. And what is this? That is having the derived allele at a frequency of 1 out of 24, 2 out of 24, 3 out of 24, and so on. So this would be what we call that a singleton. There's only one copy of the mutation. This is a doubleton. There are 2 copies of the mutation in the sample, and so on. All right. And we can then try to fit this, calculate what this frequency spectrum should be on a certain models, and then fit those models to the data. So this is an example here from some data from what was called the Seattle SNP consortium. That's very early human SNP data. And this is for a model with what you would expect under Kingman's coalescence, our standard coalescence model. You can easily calculate what this expected site frequency should be. And you can see there's sort of a discrepancy here. We got too few singletons. And then you can try to estimate a model with population growth, calculate on the population growth, and then get a much better fit. Okay. And what you could do is that the framework for fitting is that people will be using composite likelihood for fitting. That is that they would assume that incorrectly that each site is independent of each other. Why are the sites not independent of each other? Because of the correlations underlying shared correlation structure, the answers for recombination graphs. That's also what generates linkage disequilibrium. Because of this shared correlation structure among adjacent sites that will tend to a similar trees, sites are actually not independent of each other. They're correlated. So, but nonetheless, people will write down a likelihood function as if the sites are independent of each other. So the likelihood function would be the probability of seeing on a set of parameters a configuration of type J, and again, this is a configuration type 1, 2, 3, 4, 5, 6, and so on, to the power of the number of configurations like that you see of type J, and then the product over all J. And that's how we define the likelihood function. That is what's called a composite likelihood function. So again, this is the sampling probability of a SNP with site pattern J. And this is the number of SNPs with site pattern J. All right. Fortunately, so you say, okay, why are we doing this? We're taking a product of something that's not independent. So why is that okay? Well, it's what's called a composite likelihood function. Each of these functions are individual likelihood functions. You take the product of them, even though they're not independent. It's called a composite likelihood. And it has, in general, very good properties if you build statistical estimators based on functions like that. For example, we can show specifically in this case in population genetics that estimators based on this will be consistent. That is that as you get infinite amount of data, the estimates of the parameters based on this, if you use this, if you maximize this function for the parameters, the estimate of the parameters will converge to the true values of the parameters if the model is correct. So even though you ignore the correlation that are among sites, you still end up with relatively good estimators of the parameters when you do that. In some sense, what you're doing is you're throwing away some information from the correlation structure, but these individual likelihood functions are still valid functions. So basing an estimate on that is actually not a bad idea. All right. So how do you calculate this? How can you calculate for this, these site probabilities? Well, in general, what you can do is that you can calculate it as the expected time in the tree in which a mutation will induce the observed site pattern divided by the expected total tree length. And that's relatively easy to prove if you from the assumption of a Poisson process, mutation according to a Poisson process, and then taking the limit as the mutation rate going to 0, you can show that, in fact, quite easily, that is generally true and is true for any model, as long as there's no natural selection, as long as you have this decoupling between the mutation process and the genealogical process. If mutations occur, then according to a Poisson process, this will always be true. So again, this TI here, that's the time in the tree in which a mutation, in the ith tree, that a mutation would generate, if that happened on that a lineage in that time, the observed type pattern, and that is the total tree length. Probably easier to illustrate with an example. Here, move the bar up here. All right, so this is what we say it is. The probability, these probabilities that we're after. So these ones here can be calculated as the expected time in the tree, in which expected sum of, we can figure out expected sum of branch lengths in the tree, in which, if a mutation happened on those branches, it would induce this observed pattern divided by the expected total tree length. So for example here, with an example, and then it should become a bit clearer what it is. Let's say that we're interested in the pattern 3 and 2. So this is an unordered. So now we don't know which one is ancestral and which one is derived. Let's assume we don't know that. And we're interested in what's the chance, if you see a mutation, that you have 3 of one type and 2 of the other type. So that's the same as 2 and 3 here, the way I do it. Well, if a mutation happened on this lineage here, for example, then these 3 will have the derived type and these will have the ancestral type. So that's a 3 and 2 pattern. That would do it. Or you got a mutation happening here. Then these 2 will have the derived type and these 3 will have the ancestral type. So that's also a 3 and 2 pattern. Or it could have happened here. Then these 2 have the derived type and these 3 have the ancestral type. So again, that's a 3 and 2 pattern. So if mutations happen on any of these lineages in blue, you get this pattern. But if they happen on any of the lineages in red, you get singletons. And you wouldn't get this pattern. You would get a 1-4 pattern instead. So the probability of this is the expectation of 5 times the time in the tree that you have 5 lineages, because that's the length here, 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 and here. Okay, now it's calculating the expected total tree length. 4 times this time, 3 times the time in which they're free here. Let's see, it's from here to here, this time here, tau 3, and 2 times tau 2. That's expected, that's the total tree length. That's the sum of the length of all of these branches here. Right, that's capital T. And lowercase t here, well, that is the sum of the length of these branches. It's the time in the tree, it's which you have 4 lineages. That's this time here. Count it once, just for this branch. The time in the tree in which you have 3 lineages. This time here, that's for this part of that branch. Going here. And then 2 times the time in the tree in which you have 2 lineages. That's from here to here, this branch plus this branch. So if we add all of these times together, these are the coalescence time, the time in the tree which you have 5, 4, 3, and 2 lineages. You add these together, tau 4, tau 3, and 2 times 2. That gives the lengths of these lineages here. And the total tree length, the sum of the length of all the branches, is 5 times the time in which you have 5 lineages, 4 times the time in which you have 4 lineages, 3 times the time in which you have 3 lineages, and 2 times the time in which you have 2 lineages. Gives the total tree length. So that is, if you take the expectation of these, then that gives you that probability. Now, why is this useful? Well, it's useful because then all you have to do is simulate genealogies. Sometimes you can calculate this analytically. Otherwise, even for extremely complicated model, as long as you can simulate under them, you can get these expectations really fast by simulating expected coalescence times. And so you can calculate this relatively fast, typically. All right. So let's take a pause here with a question for you guys. Here's another problem. Consider the polarized side frequency spectrum. So that's now where we distinguish between ancestral and derived. We know which one is ancestral and which one is derived. So for example, in this case, if a mutation happens here, it'll create a 2 and 3 pattern that we would distinguish from a mutation here. And here, that would create a 3 and 2 pattern. We distinguish between which allele is ancestral and derived in the polarized side frequency spectrum. So consider the polarized side frequency spectrum. Under the standard, that is Kingman's coalescence with n equal 3, what is the probability of the pattern 1, 2? Meaning 1 ancestral and 2 derived allele. All right, so that's the question for you. So you might want to, when I send you out in breakout rooms, this question will disappear. So you should take this down. So you remember it's of n equal 3 leaf nodes and 3 of 3 leaf nodes. What's the chance of getting a pattern with 1 of the derived type and 2 of the ancestral type? Okay. And the idea is you want to use this formula and this type of logic. And then you need to remember what are the expected coalescence times. And let's just go back to those. We have them here. The expected time in the tree in which there are 3 lineages is a third on the coalescence scaling. And the expected time in the tree in which there's 2 lineages is 1. All right, so take this down. Time in the tree in which there are 3 lineages is a third. Time in which the 2 lineages is 1. And then take down this question. For n equal 3, what's the probability of a 1, 2 pattern? That is 1 of the derived and 2 of the ancestral. That's the question. Alrighty, so I'm going to send you out in breakout rooms again. And all breakout rooms speak Russian, except the one with Leo in, that speaks either French or English, depending on what you prefer. All right. So let me see if I can send you out in breakout rooms. I think I can. All right, please join the breakout rooms. All right, welcome back. Maybe I'll just ask if you came up with any solutions. Feel free, anybody, to unmute yourself and speak up if you have a suggestion. That's my solution. I'll show that in a second, but nobody wants to volunteer anything? All right, that's fine. I'll just go ahead. All right. So we'll have to be able to calculate that probability of that side pattern. First, we have to realize what probability do we need to calculate and how to do it. Well, we need to calculate the expected length of this branch here in a tree. So if there are 3 leaf nodes, you only have one. If you, without labeling of the leaf nodes, there's only one possible topology. So the structure of all the 3 different ones, when you label the leaf node, they all look like this, right? There's first 2 lineages that coalesce, and then they all coalesce to a final one. And no matter how the coalescent tree otherwise look, it will always have this structure. And this is a lineage that mutation have to happen on if we get 2 of one ancestral and 2 derived. That's what I write. I actually gave you I think verbally I said the opposite with one derived or 2 ancestral. I can't remember, but this is to get 1 ancestral and 2 derived. We have to have the mutation on this lineage. And the total tree length we have to then calculate first. That's this denominator here. We have to calculate this total tree length, the expectation of that. Well, that's just the expectation of the time in the tree in which you have, the total expected time of all of these lineages. So it's 3 times the time in the tree in which there are 3 lineages, which I was saying was a third, and 2 times the time in the tree in which there are 2 lineages, and the time in the tree, the expected time, the tree in which there are 2 lineages is 1, as I mentioned before. So it's 3 times a third plus 2 times 1. That's 3. That's the expected total tree length of this tree. So the sum of the length of all of these branches. The expected length of the blue edge is 1. Why is that? That is because the expected time in the tree in which there are 2 lineages is 1. And there's just 1 lineage that we're looking at. Okay, 1 edge. So all in all, it becomes the probability of having 1 ancestral and 2 derived is 1/3. And if instead you calculated 2 derived and 1 ancestral, then that would be 2/3. It has to be either one or the other, because you're calculating the probability of a pattern given that a mutation happened in that site. So there's only 2 possible patterns here, the 1-2 pattern or the 2-1 pattern. Okay, so the calculation here is relatively easy to do when first you know how to do it. There's no fancy math involved. It's just a matter of calculating, knowing what the expected length are in the tree and adding the appropriate lengths together. Okay, and for very complicated models and large site patterns, that is very, very easy to do also in a computer. Now, to really get information from multiple populations, what we want to look at is their joint sign frequency spectrum. So the distribution of allele frequencies jointly in multiple populations. So this is, for example, from a paper we wrote about 10-15 years ago, where you have Han Chinese and Tibetans, and we sequence a bunch of data for them. And these are this is the joint site frequency spectrum for the 2 populations. And the way to read this is, there's a dot here that says that the allele frequency in Tibetans is, maybe 80 or 75, 80 or something like that, 85 of the derived type. And in the Han Chinese, you found maybe 20 of the derived type. And there's just 1 SNP with that color. But if you go in here and look at something where, let's say we go in here and say 50 in Tibetans had that mutation and 40 Han Chinese had that mutation in here, we saw maybe around 50 of those or something like that. So the color code tells you how many SNPs there are of that type. So this is the joint distribution of allele frequencies in 2 samples, 1 sample from each population. So the reason why you want to do this is that this has the joint information about the relationship between the populations in it. So if you want to ask questions about the relationship between Tibetans and Han Chinese using the site frequency spectrum, this is what we're after. All right, so here's just an example of that. This is for using some very early sequencing data of European Americans and African Americans, where we had various things we were doing with that, but we had a model like this: European Americans and African Americans split up from each other. Originally, the populations, there's been population growth, there's been a bottleneck, and there's been migration between the population, and there's been a mixture. We know African Americans are in fact a mix between people with African ancestry and European ancestry. So there's a mixture there. And so you have a bunch of models with that. You have a bunch of parameters. You can, for this joint site frequency spectrum, estimate these parameters. So for example, divergence time, we estimated here at 75,000 years. Bottleneck and growth, 200-fold increase in population size since the divergence time in European Americans, strong effect of migration, and so on. And then you could look how well does the data, or does the model then fit the data. And so this is the allele frequencies simulated here is the predicted allele frequencies based on the fit, and the red is the observed. You see a good fit here for African Americans. Here's for European Americans. There's also a good fit of the relatively good fit of the distribution. So you can fit a model in this way relatively easily by calculating the site frequency spectrum. In this case, we calculated by similarly under this complicated model, simulating genealogies and just counting up the length of the branches to get the probability distribution of these joint site frequency spectra. And these are then the marginal site frequency spectra for the 2 groups that we compared. All right, and then you could test how well does the model fit the data? Can you reject the data in using predictive simulations? And in this case, we can't reject the model. We have saturated well enough that we predict the data with high accuracy and can't reject our own model in predictive simulations. All right, so that was a long time ago. Now you have very complicated models that are being estimated like this. So this is from some analysis. Laurent Escoffier, who has written this program FastSimCol2, did some years ago from a paper we wrote about the history of populations from Australia. And you see what he compares here is some Europeans and some Han Chinese, some Papuans, and 4 different Australian populations, people of Aboriginal Australians, and then estimate a number of different parameters here, effective population sizes and migration rates. And the way this is done is basically, like I explained, by simulating coalescence trees under different models, calculating the site frequency spectrum that would be induced by that jointly for all of these populations, and then using this composite likelihood function to try to maximize that. Now, this is complicated to do. This is hard to do. This is an analysis that took several different months and was to some degree very guided by the user in terms of choosing exactly which features should be in the model and other things like that. So there's still a lot of research questions about how to do this best for this kind of complicated model. I think this is one of the most complicated analysis I've ever seen published, where people have done that. Maybe the most complicated ever published. Like most complicated model with the most parameters. I think that 20 parameters or more, 25 parameters here. It's hard to estimate that many parameters. An alternative that is also being used a lot to this is to use diffusion theory. So you can, instead of using coalescence theory, you can write down some diffusion equations that specifies how allele frequencies they change forward in time. So it's the same model actually. It generates the exact same probability distribution. What you do then, you get these continuous distribution of allele frequencies in the population, and then you sample binomially from that to sample your sample, sample size of n, calculating the probability of getting, say, x of the derived type out of n. It's binomial sampling from this continuous distribution of allele frequency, and you use that then to also calculate similarly the probability of the different side patterns in the side frequency spectrum. Now, these probability densities is a set of partial differential equations you have to solve. It can be computationally very intensive. You use various standard techniques for that, for example, Crank-Nicholson, to solve those, and then after that you do this integration where you sample binomially. It's also slow and it's also difficult, but it also works and can be done, and that's sort of the alternative to that. So alternative to the simulation approach is this numerical solution of diffusion equations, and that's what you have in the program DADDY, for example. That's the most commonly used program for doing this, using the numerical solution of these partial differential equations, is to use the program DADDY. All right, so this is all complications that come in because of this this ancestor recombination graphs. And of course, really what you want to do, these are just summary statistics we're using. It gets really, really complicated, but we're just taking this distribution of allele frequencies. And remember, we ignore the entire correlation structure among different SNPs, and there's a lot of information there. And really what we would like to do is to really get at these ancestor recombination graphs in a similar way that we did for mitochondrial DNA and Y chromosome DNA, where we could use MCMC approach or other approaches where we could really integrate over the space of genealogies. We would want to do the same for ancestor recombination graphs, but it's just impossible. Also, we thought, I would say, until this paper landed. So this is, I think, is a really impressive paper written by people in Adam Siebel's group. And what they showed is they showed an MCMC approach for actually sampling ancestor recombination graph. It's slow, and it doesn't really work for more than 40, 50 sequences. It's just way too slow, and there are some problems with, but it actually does what it claims to do. It can sample ancestor recombination graphs. It's slow and takes a lot of time, but it can be done. So it is actually possible. And I think, so this is a paper that came out a few years ago. And since then, there have been a number of the other methods published for trying to estimate ancestor recombination graph. And I think the push right now in the field is to say, okay, let's see if we can do this. Let's go to actually go back and do the same thing as we did 15, 20 years ago on coalescence trees, and then do the same thing on ancestor recombination graphs. It's computationally intensive, but let's get use the fact that we have a lot of computational power to try to solve this problem, at least as an approximation. This is using really the full ancestor recombination graph using MCMC, and it's a very impressive program. You probably will be hard to make something that does exactly what they're doing, that is much faster, because there's a certain amount of computation you just have to do. But there are various ways maybe to approximate it very closely. But if you can do that, how can you then make inferences about demographic parameters? I mean, if you also had to do what I did before with MCMC for coalescence tree, then you would have to simultaneously do a integration of all the demographic parameters with the integration over the ancestor recombination graph. Maybe it's possible, but it seems unlikely to be able to work well, I think. So the push on what people are thinking about doing is instead using importance sampling. And importance sampling is really a trick that we also used in the old days, where we just used a single genealogy. So there's this problem. You have this is this Felsensteins equation. Remember, the probability of the data given the genealogy times the density of genealogies given the parameters. You want to evaluate this by, if you could, summing over all possible topologies and solving the multiple integral over all possible coalescence times. But as I mentioned, one way we could do this is instead, for example, just simulate genealogies from this density, and for all of those genealogies, evaluating this function and take the average of that. That would approximate this integral. But what we could do instead is we could simulate from another distribution. So we could take this integral and we can multiply and divide with some other density of genealogies. And so what we can do now is we can simulate from this density of genealogies, and then instead evaluating the average of this function here. So this integral here is, we can think of that as that expectation of this with respect to this density here. So if we simulate from this density and then evaluate this function here, this ratio here, for all of the genealogies we have simulated, that converges to this expectation, okay, which is this integral. All right, so now we can choose some other distributions that better approximates this. And we know it'll be more efficient if it approximates this product. So this is what's called importance sampling. It's an importance sampling approach. We're trying to find other distributions here. And that has, so this is just basically what I said. This likelihood function, this function here, can be approximated as, if you simulate from this density up here, as the sampling average of this function evaluated for genealogies simulated under this density. So this right-hand side here will converge as k goes to infinity by law of large numbers to what you have on the left-hand side. Okay, so so we use that a bunch of approaches for, instead of using MCMCs, to do to calculate likelihood function population genetics using this type of function instead, or this type of approach instead. And you can do something similar with the ancestral recombination graphs. So we can take Adam Sibbel's program here and run that once on a Kingsman's coalescence, and then use this trick to evaluate the likelihood function for other parameter values. So for other models, by using this importance sampling weighting. So we've implemented that for a couple of different methods now, but most importantly, what the only thing we publish is from actual model of selection, where we have selection in acting on a site. And basically the idea is that you sample trees, sample all this sequence of trees, that is the ancestral recombination graph. And then we evaluate, we sum over these sampled trees a certain function over that tells you something about the selection coefficient that would act in a certain site. So basically we can calculate the probability of seeing a particular genealogy if there was selection. And we evaluate that function. We use importance sampling to evaluate the likelihood function if there was for selection, even though all the trees we sampled were sampled under a neutral model. So you can write that down, and it works, and it should by in theory give the you know, by this logic, it gives as the sample size goes to infinity, it gives the exact right likelihood function. So with that, we can do things like this. So we can estimate a selection coefficient. So selection coefficient, what that is the difference in fitness between individuals that carry the mutation and individuals that do not carry the mutation. So it's how much of an advantage you get by carrying a particular mutation. That's a selection coefficient acting on a mutation. And if the selection coefficient is 0, that means that there's no selection acting. That's what we call neutrality. All right, so we simulated data under a neutral model. And this is an example, estimated then a likelihood function. We get that inferred S in average that's a little bit larger than the true S. And this is an example of a likelihood function there, and we're very close to 0. Then we simulate on when there is truly selection with a selection coefficient of 0.003. So you get a 0.3% selective advantage if you have this mutation. And then we can estimate the selection coefficient pretty well. The method will also give us the allele frequency back in time. Because we have this whole genealogical history, we get all of the information in the sample about the history of any particular mutation. It's all preserved in that ancestral recombination graph. And this is the neutral case. What we have in red is the actual trajectory the mutation went through when we simulate it. And in yellow is our inference of what the selection of what the allele frequency was back in time. And this is for the selected case. There's been a strong increase in allele frequency to the present. This is the present, this is back in time. And that is, the red is a true trajectory. And the trajectory we inferred, we overestimated for a lot of the time, a little bit the allele frequency, but we're pretty much getting the trajectory right, getting the right trajectory. So this is just for a particular example, but in general, it seems to prefer well. So now we can do something that really hasn't been possible to do before. Using ancestral recombination graph, we can quite precisely estimate selection coefficients and trajectories of mutations back in time. All right, and this is just a little sort of figure bragging about the efficacy to detect selection. You have on the y-axis, you have the true positive rate, and on the x-axis, the false positive rate. And these are under different scenarios of selection. And our method is in red, and on competing methods is in either black, yellow, or blue. And it's one of these figures that show that your method works better than other methods. So what it shows is really what it illustrates the power of using ancestral recombination graphs. We get so much more power than previous methods because we can pull in all of this information in the data from all the adjacent sites by using the ancestral recombination graph. It's all of the information is there in the data. All right, this is an example. This is for lactase, which is a very famous example of selection in humans. So this is the genetic variant that allows individuals to digest lactose as adults, so to avoid having lactose intolerance. So if you don't carry this mutation, you're not able to, when you're an adult, to digest the carbohydrate that is in milk, lactose. And you will have various forms of lactose intolerance. You can't really drink milk. Some people drink milk anyway and don't have much discomfort. Some people have a lot of discomfort drinking milk if they don't have this mutation. But if you have this mutation, you can digest this carbohydrate that is in milk as an adult, and you don't have any lactose intolerance. And what this shows is our estimate of the change in the allele frequency through time in Northern Europe, where we know there's been selection on it. And you can see there's been strong selection on it. And dairy farming was invented right around this time here. And since dairy farming, that is that you have cattle and you get milk from cattle, there's been this strong increase in the frequency of the mutation. There's been natural selection that's favored your ability to digest milk. What it means is that people that could digest milk, they were more likely to survive. And it makes a lot of sense in a cold northern winter. If you're a dairy farmer, that you're more likely to survive if you can drink milk than if you can't drink the milk. So the people that could drink the milk would survive better. Now, what you have in red here is ancient DNA. And you have either a frequency of whether they have the mutation or they don't have the mutation, the individuals, back in time. And this is then from another paper, another publication, the inference of what the trajectory is using ancient DNA, where you actually can see back in time what the allele frequency was, because you actually have individuals back in time. And you can see this sort of a proof of concept here, that we're actually able to not get exactly the same as you got from the ancient DNA. Again, we overestimate a little bit the allele frequency sometimes, but pretty darn close. We get that it has been a trajectory like this for the lactase locus. So we can estimate the allele frequencies, how that changed back in time using only modern data, and essentially get almost the same as when you have all of this ancient data. And I should say the discrepancy here can also be caused by the fact that these samples are maybe not representative really of the ancestry of these samples. These samples might have a more diverse ancestry. This might actually be true for these older samples, or for these modern samples. All right, so that was the lactase story. All right, and that was from this previous paper from Madison and Madison. All right, so here's a bunch of different trajectories that we find for different loci that might have been under selection. These are ones that have been proposed to be under selection for hair color or eye color or skin pigmentation. And we estimate trajectories and selection coefficients. Some of them are clearly under strong selection and really change the allele frequency. The KIT ligand, for example, here, strong selection. Then there's this ACEP that really, this mutation definitely has not been under selection. We see no evidence of that. There's another one, doesn't seem to have any evidence of selection, at least not recently within the last 25,000 years. One that was very interesting is this TYRP1 mutation. There's a lot of argument that this is under selection. We don't see any selection over the past 25,000 years at all for that one. But we do see from some other ones, yeah. All right, so this is another one where that's also been claimed to be under selection, OCA2. And this is an interesting one because this one has been claimed to have been transferred to humans from from Neanderthals. And so it's a Neanderthal-derived mutation, some people have argued. And we don't see much, it's also been argued it's been on a strong selection. We don't see any selection in the last 25,000 years. Are very weak. We see a little bit, like a ratio of 4.41, not very strong, not very strong selection coefficient. But when we go further back in time, we see this strong change. Okay, so in fact, what we see is that right after Neanderthal integration, there seems to have been really strong selection for the mutation, but then there hasn't been so much more recently. So the change, was relatively mild compared to many other mutations over the past 25,000 years. And this evidence we found for selection after always like the ratio was in fact for something that's much older. So we think maybe that after there was integration with Neanderthals, this is one mutation that maybe selection has been acting on. All right. So this is all I have for you for today. Let me ask if there are any questions about this or about anything else that I've been talking about today.
At the very beginning, there was a graph that I didn't understand, but how to describe it? It was when you said that with agriculture, there was a man could have more offspring, that it created a change.
Oh, sorry, are we all the way back at the coalescence trees?
No, it was when you said that with agriculture, some men could have more offspring. When you talked about Genghis Khan and all, you remember?
Oh, yeah. Okay, yeah.
And I didn't really understand what was I looking at.
Is it this figure or is it the argument about more offspring?
The figure, what does it show?
Okay, so this shows, first of all, this shows the top here shows trees that have been estimated for the Y chromosome. So phylogenies, or you can think of them as coalescence trees that's been estimated for Y chromosome and for mtDNA. So mtDNA is inherited just maternally and sit in the mitochondria, and there's no recombination in that DNA. And Y chromosome is inherited only paternally. Only men have Y chromosome, and it's inherited from father to offspring, from father to son, and there's also no recombination there. So you have a single genealogy. What this down here show are estimates of this effective population size through time in different human populations using one of these MCMC approaches for estimating effective population size.
And maybe that's... Sorry.
Go ahead.
No, it's what's effective population size? What does it mean?
So effective population size, you can think of that is if the coalescence model we're using is correct, then it's just simply the size of the population, the number of individuals. So, if we go back all the way back to this figure here, in this figure, this population size is 3, 6, 9, 10, 11. Okay, there was 11 individuals. In real populations, you have thousands, of course, but to fit it in the slide, there are 11 here. That's the population size. There are 11 individuals. An effective population size can be defined in several different ways, but it's the number of individuals in a population like this that would generate the pattern, that the amount of variability that you see in the data with respect to some statistic. That's usually how it's defined. So, for example, if you say that there could be an effective population size based on the number of segregating sites, that would be the size of a population like this, an idealized population that would generate that many, or be expected to generate that many segregating sites. So often what we are thinking about, like there are various ways to define these effective population size. And the more sort of modern way of thinking about it is in terms of actual coalescence times. So rates of coalescence. So the effective population size of a population is the size of an idealized population with the same rate of coalescence as what you see in the real population.
All right.
Okay, so
So it's supposed,
go ahead.
no, so it's supposed to indicate the population size, but for the male it doesn't. Or
Yeah,
no, I think that they get
so here, so what this tells you is that there were fewer individuals here, right? That's what it should tell you. But you have to have the same, humans produce, 50% males and 50% females. And every time you have a mating, you have to have one woman and one man, right? You can't have, you can't produce offspring in other ways. So how come that there can be this discrepancy between the two? That's a conundrum, right? That's the question. How come you can have a much lower effective population size among men than among women? And the solution, possible solution, is variance in offspring number. Because if the variance in offspring number is very large, that reduces the expected coalescence time. So you have higher rates of coalescence if there's very large variance in offspring number. That I haven't shown you. That I'm just stating here and explaining. But in fact, this model here, this coalescence model, is true for a whole range of different models with different variance in offspring number, as long as you scale by the variance in offspring number. So you can think of really what the rate of coalescence is number of individuals divided by variance in offspring number. That's one way some people think about it. I used last time when I went through more detail, I used this Fisher model that has a variance in offspring number of 1, so it all goes out in the wash. But you could use other models that have much more variance in offspring number. So you could say every generation, we choose one individual to have all the offspring randomly. Right? If you do that, you will coalesce every generation, right? The effective population size would be really small because you could keep coalescing in, every generation. Or you could have a model that's not quite as extreme. You say, let's randomly have, 3 individuals that produce all the offspring. The rest don't produce any offspring. Then again, you will have a high rate of coalescence. That's 1/3 chance of coalescing in each generation. So the variance in offspring number affects the rate of coalescence. And that's what we think is going on somehow down here. It has to do with the variance in offspring number having changed as you get a social structure where you build bigger societies and you start having cities and so on. You start also having a social structure where there's much more variance in offspring number.
all right. It's a lot more clear.
So that's the hypothesis. That's what we think. I don't know if does that answer your question?
Yes, completely. Makes it a lot more clear.
All right. So I see there's some questions in the chat. Let's see if we're running out of time. We're running out of time now, unfortunately. I should have seen that earlier. But if there are questions, maybe I'm gonna quit now because we're over time. But if you have questions to me, feel free to send me an email, and I'll be happy to answer any questions, or Vladimir can direct the questions to me. If you have questions to me, I'll be very happy to answer them. All right. So that's it for today. Thank you so much, everybody. And
thank you very much, Rasmus.